Hello, everyone. Welcome to today's live broadcast, In Vivo Imaging Toolkit, Advancing Preclinical and Translational Science in Molecular and Orthopedic Research at UCLA. I'm Christina Jewell of Labroots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by Labroots and sponsored by PerkinElmer. PerkinElmer is a global leader focused on improving the health and safety of people and the environment. Their dedicated team of 8,000 employees worldwide are passionate about providing customers with an unmatched experience as they help solve critical issues in human and environmental health. PerkinElmer's innovative detection, imaging, informatics, and service capabilities, combined with a deep market knowledge and expertise, help customers gain greater insights into their science to better protect our environment, our food supply, and health of our families. For more information, please visit www.perkinelmer.com. Before we begin, just a couple of important announcements. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button on the lower left. I would now like to introduce today's speakers. We have two speakers for you today, Jason Lee, PhD, and Nick Bernthal, MD. Speaking first today will be Dr. Lee. Dr. Lee received his PhD in DMMP under Dr. K.S. Radu and his postdoctoral training in molecular imaging at Memorial Sloan Kettering Cancer Center under Dr. Vladimir Ponomarev. With his work focuses on the development and integration of in vivo imaging assays to quantitatively assess the functional dynamics of health and disease and to study associated therapeutic interventions in preclinical models. I will now turn it over to Dr. Lee.
Hi, thank you, Christy. This is Jason, and today the presentation from my end is going to be somewhat of an overview of the types of preclinical imaging modalities that are available, but also that are mainly used at the Crump Institute here at UCLA. So I usually like to start off with this slide, which gives an overview of the different type of imaging modalities space that one can assess. Imaging in general is considered either anatomical or functional. There's also at the level of molecular imaging, which allows one to see at a much lower or much higher resolution in terms of the biological activity. So this slide gives an overview of how this imaging, how different types of imaging modalities are available and can span this space from us understanding from the genomic level, proteomic level, metabolomic level, all the way up to the anatomy. You can see that within that range, there's functional imaging, which not only allows one to see the presence of certain types of receptors or enzymes or whatnot, but actually to be able to monitor their function in real time. While there are multiple types of imaging modalities are available, it doesn't necessarily mean that they are both available in the preclinical setting in small animals, large animals, and also available in humans. There are some caveats to how they can be translatable. So I'd like to present this slide just to give an overview of what imaging modalities can easily translate to the clinic or that are already available in the clinic. For the most part, all of them can be, even though on this slide it shows that certain ones like optical is not necessarily translatable. Nowadays, that's not necessarily true. You do have some surgery, for example, that does implement optical imaging, and Nick Bernthal, who's going to be speaking next, may be able to give more insight into how that translation works. So this slide is something that I tend to present because it seems to summarize quite a bit of what it is that the Crump is able to do with the types of imaging modalities that we are well-versed in or that we have available. So you can see that, one, it spans different types of biological activity, biological questions and research. using different types of modalities. Here we mainly look at 3 different types. We have PET scanners, CT scanners, and optical scanners. And then on the lower left corner, you could also see some high-resolution metabolic assays that we can perform, and I'll go into that a little bit later. Generally speaking, there are certain types of parameters that one wants to consider when they're looking at imaging modalities to determine which one best fits their research projects. For the most part, most users would look into how sensitive are these imaging modalities for detecting the structures, the anatomy, the activity or function that they're looking for. And then also the spatial resolution. Is it able to detect single cells, or are we looking at larger anatomy like different types of organs? And it's not one or the other. Usually one wants to look at both sensitivity and resolution. Ideally, of course, you want to have the highest resolution and the highest sensitivity. But based on this chart, one can see that it's not necessarily available for all different types of modalities. There are pluses and minuses for each one. So the key is not necessarily to find the one with both high sensitivity and high resolution. Ideally, that would be the case. But the key is really to find which one fits the research question of interest, and to determine if sensitivity is more important, resolution is more important, or is there a compromise between the two that one can select. So the ones that we tend to have here at UCLA does span everything that you see here. Even in the clinic, we have SPECT scanners and PET scanners, CT, MR, and optical. In the preclinical setting at the Crump, we focus mostly on PET, CT, and optical imaging, and then to a certain degree, we also collaborate with another center on campus for MR. So generally speaking, we do have all these modalities available to our users. And this chart is just another explanation of the different type of parameters that one would like to consider. I put them in the order of different types of sensitivity, and they're all relative to MR. So if we look at relative sensitivity on the right-hand side, MR is 1, and the higher the number, the more sensitive some of these assays are. And then we also have the spatial and temporal resolution. Those you have higher spatial resolution when one wants to look at anatomical structures, because for the most part, those do not require additional types of agents that need to be injected or different types of detectors. And so, for example, MR and X-ray CT, one can see has some of the highest spatial resolution. Even though here it says 100 microns, they actually can even go down to sub-micron levels like 50 nanometers, for example. But that's also, relatively speaking, depending on certain types of reconstruction methods or algorithms that some of these scanners would use. This is another summary, the last summary slide that I have here for the different types of imaging modalities. For the most part, it's to give a sense of how do I select which imaging modality for what type of questions or process or experiments that I have. So generally speaking, optical imaging tends to be the most high throughput, either when it's in vitro or in vivo, simply because of the lack of complexity of it. One just detects light emitted from the cells of interest or the organs of interest. In terms of sensitivity, PET does have a slightly higher sensitivity. Optical technically does have higher sensitivity, but because of caveats of detecting light, in general, PET does have higher sensitivity. The last parameter there is this potential for clinical translation, even though here it kind of mentions that PET is the most clinically translatable, MR not being so much, but that's not necessarily true per se, depending on the situation. And for the most part, all 3 of these modalities can be translatable to the clinic in one form or another. So going to how we do imaging here at Crump, this is just a summary slide of all the imaging modalities, equipment that we offer at the center. In the upper left, we have 3 different types of PET scanners, the Siemens Indion, the Siemens Focus, but then now we also acquired just recently the PerkinElmer Genesis Genesis 8 micropet and CD combined system. For us, that's actually been one of the more high throughput instruments that allow us to do many studies very quickly. On the right-hand side, we also have the IVIS Lumina from PerkinElmer and a CRI fluorescence scanner. So the fluorescence scanner is simply for imaging animals at different levels of wavelengths for fluorescent agents, whereas with the IVIS Lumina, we're capable of doing both fluorescence and bioluminescence. And I'll go into the bioluminescence a little more in a second. On the bottom panels, we have the Elixis automated synthesizer. That's actually related to PET imaging, where one has to synthesize the radioisotopes necessarily for measuring different types of biological activity. We have an in-house, what we call a Crump micro-CT that was built in-house for us to do relatively high-resolution small animal imaging. And then the bottom right two types of equipment is for ex vivo analysis of some of these assays. The automated gamma counter allows us to look at biodistribution on tissue-to-tissue level. And the macro and microkeratomes allow us to do certain assays like autoradiography, which I will show a demonstration of that in a second as well. And this slide is definitely not a summary of all the different types of publications that we have done here at Crump. It's just giving 4 examples of how imaging can be used for functional analysis as well as anatomical here at Crump and at UCLA. The top one is looking at different types of PET tracers, PET isotopes that were used to look at different types of biology. The upper right-hand side is looking at immunology, cell trafficking of immune cells. And the bottom left is also looking at cell trafficking of immune cells, but using optical imaging. And then the bottom right is actually looking at pharmacodynamics, the ability of imaging to allow one to see the effects of drugs in real time. So going to the first one, I'm going to split this into 3 sections. The first one is on optical imaging, mostly focusing on bioluminescence imaging. The idea is very similar to fluorescence imaging, so I think it would cover both. So just a general overview of bioluminescence itself. Essentially, a user expresses what's called a reporter gene. In this case, if you look on the left-hand side, it's called firefly luciferase. And the way light is produced is that firefly luciferase expression alone in the cell doesn't produce any kind of signal. It is its interaction with a substrate, in this case called luciferin. The interaction between luciferin and firefly luciferase is what creates light emissions. And that's essentially what's being detected by the CCD cameras, for example, the IVIS Lumina that I mentioned earlier. And so with bioluminescence imaging, it allows one to look at these different types of luciferases. And this panel is a good summary of what type of luciferases are available in the research community. I also put a link at the bottom if anyone is interested in purchasing or acquiring some of these plasmids that have these different reporter genes in there. The table here is mainly to summarize that there are quite a bit of luciferases available, and that one can choose which one based on the several parameters that it has. The most important of which is probably the kinetics. One can see that it says glow kinetics and flash kinetics. The main difference between the two is how long, once the interaction occurs between the substrate and the luciferase, how long that light stays present for the camera to detect it. Generally speaking, firefly luciferase has been used quite a bit because it undergoes what's called glow kinetics. And so the moment a substrate is injected, that signal stays around on the order of minutes, let's say, for example, 10 to 15 to 20 minutes. And that makes it very easy for users to do high throughput studies and to quickly go through several animals at a time. Whereas with flash kinetics, it generally doesn't do high throughput simply because the moment the substrate is injected, the signal goes through exponential decay, and so acquisition of that light needs to be done as soon as possible, and also very consistently from day to day if one is doing longitudinal imaging on the same animal. And The right-hand panel, or the right-hand peak emissions is also important. Most people tend to not worry so much about that, but wavelength emissions, each of these luciferase reactions do have different wavelength emission patterns. And depending on how one wants to use it, one can take advantage of the different emissions to do multispectral imaging. For example, if you had two cell populations in the same animal, one can be expressing a certain report with 465 nanometer wavelengths, and another one can express something at a much higher wavelength. And the scanners themselves have spectral unmixing properties that allow you to actually be able to detect both at the same time. Of course, that assumes that you also inject both different types of substrates to be able to separate them as well. So these are two parameters that one tends to look at when doing optical imaging. Light itself does get absorbed through different tissues and different media that's inside the body. And you can see that it's dependent on the wavelength emissions as well. While it looks like the least absorption is probably around the 700 nanometer range, that doesn't necessarily mean that it's the only wavelength at which one can actually acquire data. It simply means that when assessing the results, one needs to take that into account. Or if one is trying to optimize the sensitivity of your reporter. Similarly, light, like any other at different wavelengths, does get scattered through the tissue, and that is also dependent on the wavelength. The higher the wavelength, the less scattered. And with less scatter, that means that the signal does get to the CCD camera, which less interaction through the tissue before. And so combining both absorption and scattering when one is studying in vivo imaging, to take these two into account can really help in optimizing the project. So this is just a summary slide of different types of imaging that you can do with optical imaging. This is actually only bioluminescence, but fluorescence is very similar. If you're familiar with flow cytometry and choosing different types of fluorescence agents, it's the same thing in vivo. The only difference is now you're imaging within a live animal. And so if these cells are expressing the different types of luciferases, you can actually look at trafficking patterns. So you can, on the left-hand panel, is actually a study of immune cell trafficking over time. The top and bottom is just differentiating between different types of treatments that the animal underwent. And the circles there are showing the expression or the trafficking of the T cells to certain lymph nodes. You can also do lung metastasis models and localizing that to specific regions. So you can see it's very specific and very high, generally very high resolution for you to actually see where the organs are, their localizations of those organs. Here you can clearly see that these are two different lobes of the heart where these cancer cells have metastasized to. And a lot of users tend to do proliferation studies, expressing reporter genes in tumor cells, implanting those tumor cells, and using imaging to watch the growth of those tumors. The presence of a signal is also important because it actually says that these cells are functional. They are expressing these luciferases and generating these proteins. And so it's also good to look at whether or not your tumor cells are viable within the animal, or any type of cells that you're interested in. The other modality that we do here, both by itself and also in combination with other imaging modalities like PET, is high-resolution micro-CT. CT basically uses X-rays in 360-degree projections to allow us to actually look at a three-dimensional tomographic image of in this case, the animal's body, the mouse's body. And these mice are relatively small. I would say they're probably a couple centimeters in length, a couple centimeters, maybe 2-3 centimeters in width. But you can really get high-resolution anatomical structures simply from imaging using micro-CT. The biggest difference between micro-CT and MR, both of which can allow you to look at anatomy, is that MR is very good for soft tissue, whereas micro-CT is very good for denser tissue. So you can clearly see with micro-CT the bone structure is very apparent. But if you look on the middle panel, but also the right panel, which has a much higher field of view, you can inject contrast agents that have different X-ray density effects, and you can actually look at the specific organs of interest. So in this case, on the right-hand side, you can actually see the spleen on the left side of that animal because we injected an iodine contrast. So Christy, if you can pull up the media video of mouse whole body contrast CT. Okay, thank you. Can you also pull up the mouse vascular micro-CT? Thank you very much, Christy. So I hope you got a sense from looking at those two videos what the capabilities are, or at least be able to appreciate the capabilities of imaging CT alone. Looking at anatomy alone, there's quite a bit of information that you can get out of just simply looking at structure. There are users, for example, that are interested in looking at vascular calcification of different tissues, and CT would be fantastic for that because of that level of density coming from these calcified regions. But also, you can inject the contrast, as I mentioned, and actually be able to get the vascular structures in the whole animal. All these imaging modalities are done on whole body level, so we simply image for our scanners, we image for about 2 minutes, and we are able to get these kind of resolutions and these kind of field of views of the whole animal. So for micro-CT, we also combine it with our PET imaging, and I'll explain to you in a second what PET imaging is, to allow us to look at both functional and also localization of where these functional signals are coming from. And micro-CT allows us to localize that based on anatomy. So at the Crump Institute, our bread and butter, I would say, is positron emission tomography, which is PET imaging, using radio-labeled tracers. These tracers range from small molecules, which are from traditional pharmacology, drugs of interest, biological molecules inside the body, for example, glucose, aptamers, peptides, and also proteins, and a lot of which currently are focused on antibodies, minibodies, diabodies. So the biological space is pretty large in terms of what PET imaging can allow us to see, depending on the tracers that we radio label. So radio labeling simply means adding, in this case, what you can see on the small molecules, an F18, a fluorine-18 isotope that emits gamma rays, and by allowing us to see these gamma rays with these detectors, we're able to actually see the biodistribution of these different types of molecules. And depending on the molecules that you choose, you can see what's called functional imaging, or you can also see what's called reporter imaging, or localization of certain proteins or receptors in the body in real time. So a good example that most people are probably familiar with, and that is generally done in the clinic, is FDG imaging. FDG is an analog of glucose that has been radiolabeled with the F18 isotope, radioisotope. On the left-hand panel is an animal that we injected with FDG and underwent PET-CT imaging. So the color scale and the color patterns that you see on that image is actually coming from the FDG itself and its biodistribution. What is interesting is that because it's an analog of glucose, when you look at that image, the PET image, you're actually looking at which tissues are highly glycolytic, which tissues are actually metabolizing that glucose or that glucose analog with high avidity. So you can see BF there suggests it's brown fat, and this animal happened to not be under a warm condition, and so the brown fat was highly glycolytic, highly active to maintain the heating of the animal itself. The heart, which is right below the BF, MY for myocardium, you can also see that's also very avid for FDG, and the reason for that is that the heart itself is a muscle that is taking up a lot of glucose in order for it to function. And that lends us to look at the next type of modality, which is complementary to the PET, what we call autoradiography. Essentially, it's the same thing as the PET imaging. The only difference is that it's an ex vivo assay. So if you look at the gray images there, that's the same animal that's been injected with FDG, but we do autoradiography, basically expose slices of these animals to film that's sensitive to radiation, and that allows us to have a much higher resolution of these biodistributions of, in this case, the FDG tracer. And I zoomed into the heart just to show you that the FDG signal really is coming from the myocardium. There are plenty of other tracers that have been developed. At UCLA, one of the investigators developed a probe called FAC, which is an analog of a nucleoside compound, deoxycytidine. And because it's a substrate for the kinase in the nucleoside metabolism pathway, its PET image suggests that it's actually measuring in real time the function of that pathway. And what they demonstrated is that indeed, if you have so DCK is the kinase that's specific for FAC, that is required for FAC to be metabolized in each of these tissues. And so they imaged it with a DCK knockout mouse, which lacks that DCK enzyme, and clearly it demonstrates the specificity of the FAC probe for imaging DCK-dependent nucleoside metabolism. If you also look at the DCK wild-type mouse, you can see that nucleoside metabolism is very high in immune organs, the bone marrow, the spleen, the thymus. And so this suggests that this tracer can allow you to measure the function of different immune cells, or the presence of different immune cells and their function. But it's also useful for other applications. FAC allows you to see the function of the immune cells, but it could also allow you to measure the pharmacodynamics of drugs that target these enzymes of interest. So in this case, because FAC is specific to the DCK enzyme, by developing a DCK inhibitor and using the FAC probe, you can actually image in the animal the effects of the drug, the effects of the inhibition of DCK. And so here it's simply doing FAC imaging across different time points, pre and post treatment of the DCK inhibitor. And you can clearly see that the DCK inhibitor has a very short therapeutic, half-life in the order of hours. And this is very useful to be able to determine what type of drugs and how successful some of these drugs can be. And this can also be translated to the clinic, that if this inhibitor was clinically relevant, patients can be scanned with FAC after treatment to see whether or not target was hit. The other type of imaging tracers for PET would be what's called immuno-PET, and these take advantage of different types of antibodies, mini-bodies, diabody structures, radiolabeling them and injecting into the animals to look at their targeting of surface receptors, surface antigens. This is just a summary slide to show the different sizes of these proteins and how they distribute through the body. You can see from the graphs themselves that the smaller the protein, the smaller these antibodies or antibody analogs, the faster they clear through the body. But it also shows that the better that they may bind to the surface antigens. And finally, I'd like to end with imaging of immunotherapy, which I think is a very hot topic nowadays. So it'd be good to kind of go over how imaging, especially PET imaging, can be integrated into immunotherapy. This is simply a schematic of how adoptive cell transfer immunotherapy works. Essentially, a donor could be the same mouse, but a donor that has antigen-specific T lymphocytes against, for example here, the T cell tumors of interest. Those cells are cultured ex vivo, and during that, because they're cultured ex vivo, one can actually take advantage of that stage and introduce the reporter gene. And by introducing the reporter gene, that allows you to subsequently be able to monitor the biodistribution of these cells in vivo once they're injected into the animal, into the patient. And so here's an example of applying optical imaging, in this case bioluminescence imaging, into an animal that underwent a full treatment of adoptive T cell transfer. You can monitor this animal from the day after injection all the way to when the population has expanded throughout the body. But not only that, you can also see the localization of where these cells actually traffic to, whether or not they're hitting target. In this case, this mouse has a brain tumor and T cells that are targeting antigen in these brain tumors. And you can clearly see where those white arrows are, that indeed the T cells are active and that they are targeting the tumor cells of interest in the brain themselves. But additionally, you also see that T cells are developing in other regions of the body, in other lymph organs, in particular bone marrow and spleen. And earlier we mentioned using antibodies. So because these T cells also express surface receptor, surface antigens, CD8, CD4s, we can also develop antibodies that target each of these subpopulations and use these antibodies as imaging tracers to detect these cells in vivo. So Richard Tabari here has developed a CD4-specific immunopet agent and a CD8-specific immunopet agent. And if you inject that into your adoptive cell transfer model, you could actually detect subpopulations of T cells in the body. And this just summarizes the different types of nuclear reporter genes for PET imaging. You can see that depending on what these adoptively transferred cells are transduced with, you can use different tracers, different PET tracers, to detect these populations in vivo, each one having their own caveats in terms of sensitivity, ability to detect the cells in vivo, their enzymatic or receptor-based or transporter-based. But essentially, any of these should be able to allow you to look at the cells in vivo once they get adoptively transferred into the animal. And so a good example of that is here, where on day 0, these cells that express the reporter genes are injected into the animal themselves that was bearing a tumor. And you can use PET imaging, a reporter-based PET imaging, to look at the trafficking patterns of these lymphocytes and whether or not they hit the target, in this case, a subcutaneous tumor. But also you can see that they localize to different lymph nodes, draining lymph nodes, and you cannot see it here, but also the spleen. And I hope I gave you an overview here of the different type of imaging modalities that are available both here at Crump itself, but also in many other institutes. And the main message is that imaging is an indispensable tool in order to understand biology and understanding biology within the animal itself. So with that, I'd like to put it back to Christy.
Thank you, Dr. Lee. And our next speaker is Dr. Nick Bernthal. Dr. Bernthal is an orthopedic oncologist who has been on the UCLA faculty since 2013. He graduated magna cum laude and Phi Beta Kappa from Princeton University and received Alpha Omega Alpha honors from Cornell University Medical School. He did his residency at UCLA in orthopedic surgery and did fellowships in orthopedic research and musculoskeletal oncology at UCLA and the Huntsman Cancer Institute, respectively. His clinical interests are bone and soft tissue tumors, and his laboratory is pioneering new implant coatings to prevent surgical infections. I will now turn it over to Dr. Bernthal for his presentation. Dr. Bernthal?
Thanks very much, Christy and Jason. Thanks for the great overview. That was really terrific. So my talk's going to be a little bit different. I'm going to be a little more specific into using some of these preclinical models to answer some of the questions we see as clinicians, and that's one of the nice things is being a clinician scientist. So basically, I just want to start out. I got very interested in orthopedic implant infections and using some of the tools that Dr. Lee described earlier at the Crump to look into these. And for those of you who aren't in the orthopedic community, this is a huge issue both for our patients and for our health system. Orthopedic implant infections alone cost the United States over $8 billion of additional healthcare spending every year. For me, more personally, I'm an orthopedic oncologist, so I take care of mostly young people with bone tumors, and they're on chemotherapy at high risk of infection, and if they get their implant infected, they're often at risk of losing both the limb and their life from these infections. So this is what one of these implants that we put in looks like, taking a whole femur out and replacing it with an endoprosthesis. And once it gets infected, you can see you've lost the game here, and you really can't help your patients anymore. So when we looked back in 2010-11 at animal models of implant infection to answer these questions, we realized that most of them were histology-based. They required large numbers of animals. You get one data point from every animal. So our initial goal was just to develop a mouse model that would allow us to longitudinally track this and to understand the phenomenon. This is our original design of our model, and basically it's a very simple surgical model. A K-wire is placed into the femur, and then the joint is inoculated with bioluminescent bacteria. We genetically engineer these mice so that both their neutrophils and macrophage give off fluorescent wavelength, and that way we can track with these IVUS imaging systems both the infection with bioluminescence and the host immune response with fluorescence in real time. These are pictures of the surgery. We open the knee joint, we ream the knee, place the pin, and then inoculate, close up, and that's a postoperative radiograph in H there. And you can see here, these are the micro CT pictures, and you can see good placement of the implant representing the model. We do have a media file here to show that if it's easily accessible. If not, we'll just go forward and we'll get to the media files maybe at the end. So thanks very much. So we look at these models with the IVUS imaging system. We also used histology early on in variable pressure scanning electron microscopy, and these are actually ways to validate the imaging modality early on. And now that it's been so consistent, we've really stopped doing a lot of the histologic endpoints. But when you look at this, our initial model taught us a lot, and you need 5 million colony-forming units to create an infection in the skin and sub-Q of a mouse that the immune system doesn't just wipe out. You can see here with just 500 colony-forming units, down at the this last barrel line here, you create an infection that goes out to 10 days without issue. And we've tracked this out to 90 days. This stays consistent. So with just 500 bacteria, you do create an infection that can be tracked. Taking the pins out and sonicating them, doing colony counts, we saw that our R-squared was almost 1, suggesting that the light did correlate very closely with bacterial counts. And then we started looking at the host response, and you can see over on this right panel, the bottom line is an uninfected mouse. You see there's an immune response here down on this bottom curve to the infection, but is down to baseline by day 5, whereas in an infected mouse it stays up because this infection is recruiting neutrophils with the postoperative inflammation being increased. And that leads to the clinical problem, which I'll show in a minute. Early histology showed neutrophil recruitment in an infected mouse, very little in an uninfected mouse. And then variable pressure scanning electron microscopy showed us biofilm formation on infected pins, whereas you see sheet metal on the uninfected pins that are clean, no bacteria is formed on them. We used the quantum FX to look at the cross-sectional analysis, and these were really beautiful images that helped us understand the phenomenon. If you look on the left panel where it's Staph aureus infected implants, you see these little pins these little pits indicated by the red arrows. This is osteolysis. This is what our patients complain of. If the implant is infected, it's honestly the problem for the patient is that it induces an osteoclastic process of eating bone away, and the implant bone interface starts to break down. Then you have a loose implant. You can imagine walking around on an implant that's rattling along inside your bone is not a comfortable feeling. So we overlay everything here with these bioluminescence and the micro CT, and you can see in this media clip exactly what this looks like. Great, thanks very much. So we're gonna, and then we've developed this with multiple models and looking into the spine. We as surgeons, every surgeon thinks they're very important and special, and so the spine guys said, hey, make us a model, and the knee guys wanted their specific model, and everyone wants their model, and so we decided to make a spine model here, and I incorrectly thought this wouldn't add much to our understanding, since I've realized that the anatomic location is really critical, and I'll show a bit of that later. But same idea, surgical approach, and then inoculate with bioluminescent bacteria. And here with this model, you can see surgical technique in these pictures, same as before, post-operative imaging. This is just a basic X-ray image. And then we look at the bioluminescence, and we can track this. This one's out to 35 days. You see an infection at 1E2, just 100 bacteria are clearing, whereas 1,000 bacteria are up and creating a chronic infection. 1E3 and 1E4 both stayed chronic out to 35 days, but 1E4 showed skin breakdown, so we selected the 1E3 dose of this Xenogen 36, so it's XEN36 strain of Staph aureus. Again, CFUs, and here is that fluorescence tracking the neutrophils. An infected mouse, it stays high, control it goes down. So I just want to spend 5 minutes here showing you some of the applications, and some of this is preliminary data, some of it is published data, but if you look at some of the questions these models answer for us. So when we get our implants infected, we always debate with our infectious disease colleagues, what antibiotic do we treat these patients with? So we decided our model could answer this. We do the mouse surgery and inoculate the animals, and then we try 2 very common protocols, vancomycin alone or vancomycin plus rifampin, as well as some controls. We gave them IV antibiotics for 7 days, and then we watched the infection. So if you look here at day 7, sorry, I'm going to get an arrow for you. At day 7 here, we start giving the IV antibiotics, and you see the combined group, vancomycin plus rifampin, goes dramatically down. And if you used a histology-based assay, you'd say this is the right answer. You'd be done at this time point, and you'd feel really good about your answer. But the story gets a little more complicated when you start following these things out longitudinally. By day 21, both the vancomycin group and the control group start to come down. So the immune system alone is starting to clean up this infection. Vancomycin and the combined group both do have fairly equivalent results. Follow it out to 35 days, and you see that there's another bounce here, that you've cleaned out the soft tissue infection, but you're getting an above baseline signal at the spine regardless of which group you use here, and that's the biofilm. That's the bacterial infection on the implant that's not clearing irrespective of your treatment dose. When we take the pins out and culture them, sonicate and culture, we get almost the identical number of CFUs off the implant, whether we treated or didn't treat. And this has profound implication. I mean, this really validates our idea that the metal has to come out if it gets infected, because no matter what we treat it with, we end up with that biofilm formation on the pin. We work a lot on implant coatings. We've designed a smart antimicrobial implant coating that recognizes bacteria and elutes out antibiotic in the face of a bacterial trigger. It uses a pH trigger caused by the bacteria to release antibiotics when it's challenged. This is my collaborator, Tatiana Segura, who really did the bulk of the work on this design. And if you look here, here's what the pins look like. Here's all in vitro testing, classic in vitro testing, and then assaying it against a more acidic environment. And you see that it dumps out antibiotic against an acidic environment here with the pH change. If you then look, now the power is, let's go back to the model and see if it works. So a coated pin in blue up top shows that same curve we've seen all along. That's an infection. The immune system kicks in around day 7 and brings it down, but it stays well above baseline going out to 42 days, whereas if you have the PEG coating with the antibiotic in it, the tigecycline or vancomycin, you're down below baseline detection. The colony-forming units validate this, that the coating seems to really work to protect the implant. And x-rays for us orthopedists, we like to see the clinical problem. And you see up top with no antibiotic in it, you get what's called osteolysis here. And this is a completely loose pin in the femoral canal, whereas in the other two that have antibiotic in them, you don't have, you don't have any loosening. Novel antimicrobials, we're starting to work in this field and develop novel antimicrobials. Gerard Wong and Andrea Casco are two collaborators who work on taking antibiotics and giving them an antimicrobial effect. This is really a very nice chemistry that they work on here, and you can see they take tobramycin, a very common antibiotic we use, and adding a group here, a functional group here to the end, they make what's called pentobra. And in vitro testing shows that it has great efficacy against Staph aureus and P. acnes, two of the bugs we see commonly. So we've added that into our coating now and are testing that in the animal model to see, using the animal model as that assay to see how successful this will be for our patients. Finally, I just want to show how we can use these imaging modalities to look at the host. And if we flip the script here and start looking at the host, as surgeons, we oftentimes try and talk our patients into quit smoking and lose weight and control their diabetes. We don't have very much success at it. So one of the ideas we had was to look at giving a boost of 25 vitamin D prior to surgery, as we know this is an immune modulator through this cathelicidin pathway. And here's some of the previous data, published data, looking at this 25D-driven immunity. But for us, we wanted to ask a really simple question. Can we prime the host immune system for surgery? And so we developed a protocol that took lysozyme EGFP mice. These are those that have the neutrophils that are fluorescent. We made one group vitamin D sufficient, normal vitamin D levels. Two groups were vitamin D deficient, and then one group got a rescue dose of vitamin D just prior to surgery. When we look at this, we saw really, first we drew blood levels to make sure we were in fact depleting them. The deficient group stayed deficient throughout the 14 days of the experiment. The sufficient group was sufficient throughout, and the rescue group did in fact get rescued. And if you look here, these are profound results. With a single dose of 25D prior to surgery, our rescue group matched our sufficient group. We saw a logarithmic difference, statistically significant, and this is about a 5 times different in infection in deficient and sufficient mice, with deficient mice having a higher infection burden, and this is rescuable, which just has such profound implications for our patients. If we can give them a single dose of vitamin D, a well-tolerated FDA-approved vitamin prior to surgery, if we can really cut infection rates this dramatically, this is maybe an easy solution to make a real dent in a huge problem. We do the CFUs, and we saw matching again, far fewer bacteria when we had sufficient or rescue mice that had vitamin D levels up to normal. And you see again the fluorescence, the neutrophils here. The deficient mice had far greater neutrophil influx, suggesting that this vitamin D effect is not driven through the neutrophils, but rather when you look here and you do a monocyte or macrophage assay, you see the deficient group was unable to muster the same macrophage response. So we're working on this pathway suggestive of a macrophage-driven response. We have two parallel trials going on now in both mouse and human patients, and you see how quickly you can translate these results into the clinic. The last thing I just want to show quickly is some tools of intraoperative imaging, and some of our collaborators in the Netherlands have published this data that shows using an IV injection of a fluorescent probe, you can potentially tag bacterial cells in vivo. If we could do this and our bioluminescence would be used as the control, if the fluorescence matches the bioluminescence, suddenly now we could see the bacteria in the operating room in our patients. So what we do currently is take out everything, every bit of metal someone has. If we assume an infection, everything comes out. This is a disaster for our patients. If we could actually see the bacteria and what parts of the implants were infected, we could replace just the infected part. We could potentially clean it and re-image and see if we actually got the bacteria off. So this is all a tool that I think is coming down the pipeline. You can see here with a Vanc 800 probe, and this is a fluorescent probe in the middle, it matches our bioluminescence control at 6 hours, suggesting that we can in fact see the bacteria, whereas at 24 hours the fluorescence is gone. And just conceptually, the idea here is that bioluminescence bacteria are genetically engineered bacteria, so they're great for study but not for our patients. If you had a fluorescent probe that tags actual in vivo bacteria, we could use this probe in our patients who don't have bioluminescent bacteria but have naturally occurring bacteria. Here are some more images, and a mouse that had no bioluminescence here, suggesting no bacteria. We had no fluorescent signal as well. So this is really promising and coming down the pipeline. We've opened the skin and opened the muscle, and imaging in real time lets us continue to see the bacteria as we try and clean it. So I know I went through this pretty quickly, but I just want to make the point of that I do think we can decrease infections. This is a huge clinical problem for our patients, for our health system, and that using these model systems as a fast screen, we really can decide what tools, what coatings work, what vitamin manipulations work, what antibiotics work, and this is the way we can translate these things into the clinic at an efficient rate without using histology. Thanks very much. Here is some of the funding and a lot of people to thank for all this work.
Thank you so much, Dr. Bernthal. Now, before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can, and if we don't get to your question, we will answer those via email after the presentation. Now, our first question is for Dr. Bernthal. Dr. Bernthal, what are the severe outcomes from a chronic infection caused by an implant?
So, yeah, the severe, sorry, can you just repeat, what are the severe outcomes? From a chronic infection caused by an implant. Yeah, so when an implant gets infected, what happens is the bacteria forms a biofilm on the implant and sets up shop, and we really don't think it's treatable. So what happens to the patient then is over time, it recruits inflammation, and these osteoclasts, which are the host cells that eat bone, they come in and they start to eat away the bone around the implant, which some of those axial pictures on the CT scan showed. And then the implant becomes loose, and when the implant becomes loose, it starts to become painful. So that's the mechanical symptom, is just pain and an inability to bear weight. So a lot of these implants are what someone's walking on, a knee replacement or a hip replacement, and they become loose and painful. But systemically, these also become nituses of systemic infection. So for my patients who are oncology patients with implants, these infections can often be very much life-threatening, and patients start to get systemic infections, which can absolutely be devastating.
Thank you, Dr. Bernthal. I actually have another question for you. Do specific models for each orthopedic procedure need to be established, or is one model of an implant in bone sufficiently representative?
So this was a question that I was absolutely wrong on. When we made our first model, I sort of said, all right, we've got an implant in bone model. This should be sufficient. But what we found is the host environment is totally different. So in the spine, where you have a lot of muscle surrounding the implant, you actually get a much more robust immune response as opposed to the knee that's synovium is surrounding the knee joint, and neutrophil influx is a lot less. So implants, coatings, antibiotics, all of them really do behave far differently in different anatomic locations. So I've become much more of a believer that you need to model the specific test question you're worried about for your patients.
Dr. Bernthal, have you looked at any knockout mice to better understand pathways of host response?
Yeah, we have. We've used this a lot, and I didn't show that data because of time, but we've knocked out IL-1 pathways and toll pathways and really gotten a good sense of, at least big picture, what the host response is to infection. Because really the fundamental question, as good as we think we are as surgeons in keeping a sterile environment, there's bacteria in the operating room. And when we think about the fact that if we talk about the 1% of patients who get these major infections, 99% didn't get the infection, so there's something the host response is doing. So these pathways and knockout mice are really a powerful tool.
Thank you very much, Dr. Bernthal. Now, our next question is for Dr. Lee. Dr. Lee, what instrument parameters can I adjust to increase the sensitivity of detection in luciferin imaging using Lumina 3? Will it increase the dosage of luciferin, increase the sensitivity of detection? This is question number 4.
So the luciferin itself, the concept of the optical imaging is that the luciferin is already injected at a sufficiently high level to saturate the system. So increasing the luciferin dosing may provide a little bit of increase in signal, but generally it should not do too much. What you can do is actually on the software itself, there are 3 parameters that can be adjusted. One is the exposure time, the amount of time that the camera is left open to acquire the photons that are being emitted to the camera. The longer you expose it, the more signal that you get, and the better signal to background that you should be getting. The other 2, which are generally less manipulated, are the f-stop, which defines how large the opening of the lens is to acquire that data. The smaller the opening, the less signal you're acquiring. So if you leave it wide open, then you're capturing as much signal as possible. Of course, the smaller the opening, the better the resolution is, because you're actually now doing somewhat of a pinhole imaging. And then the last one is what's called binning, and that's just how the pixels are combined together to give you higher sensitivity in a certain region of view.
Dr. Lee, what kind of contrast agents can you inject in high-resolution micro-CT?
So micro-CT there's many different types of chemical compounds that you can inject. Generally speaking, iodine-based contrast and gold nanoparticles are some of the main ones that are being sold commercially. Relatively inexpensive. Iodine seems to work really well. So the image or the video that was shown earlier for vascular CT imaging to look at the vasculature, the circulation system, that was using a compound called OmniPac. And depending on how you inject it, that will define how well that contrast works. So for that image, to get the high contrast for the vascular bed, because these contrasts will biodistribute and clear out the system very quickly, you would want to do something like a CT imaging during the infusion of that contrast.
We have so many great questions coming in. Let's see. Dr. Lee, what was the X-ray dosage used for the mouse vasculature CT image?
So the scanner that we have is relatively low in terms of dosage, radioactive dosage. I think it was is determined to be about, let's see, 40 to 50 milligrays, so relatively low.
What is the largest animal species with which you have experience, and which anesthesia agent do you use most frequently?
So traditionally, nowadays, it's using the anesthesia is called isoflurane mix. It's about 2% that we use for mice and sometimes rats, mixed in oxygen or air. So generally speaking, most institutes nowadays either use isoflurane through a vaporizing system, or you can also do injectable anesthetics.
Here's a great question.
Oh, sorry, and the second part of the question was
go
about
ahead.
the type of animals. So most of what we do is in mice. A lot of our scanners here are able to handle mice, rats, but also they have been used because of the large bore size of the imaging scanner. We could also do, or there have been done primates, but I personally do not do primates myself. We do mostly mice and rats.
We have an audience member who submitted a question saying, I am imaging intracranial tumors after implantation. Will the skin glue on the mouse head affect the luciferin imaging accuracy?
So potentially anything that interferes between, that has an interface between where the signal is coming from and the camera itself potentially can interfere with the signal. Some glues should not have any effect or not some dramatic effect on the signal, but that is something that you would need to actually assess. One thing you can do is simply seed some cells in a plate and then image that using the bioluminescence imaging or fluorescence, depending what you're looking at. And then maybe put a layer of glue on top of that and see if that same signal has diminished simply because of the presence of the glue, just to validate how much it attenuates the signal.
Dr. Lee, with many imaging modalities, how does one select the imaging modality of choice?
So that actually comes down to what is the basic research question that's trying to be addressed using these in vivo models. I mentioned that there's different types of imaging modalities, and each one offers different types of information. Functional imaging is if you're interested in looking at what happens to a cell, what is the biological activity of that cell during the imaging process. So we showed FDG, and using the FDG PET imaging, we're able to actually see the glycolytic metabolism, glycolysis, in the animal tissues or in the tumors or cells of interest. If you're interested in looking at the structures, or for example, if you want to look at how different layers of fat are being affected by treatment, then you can actually use something as simple as anatomical CT imaging and analyzing the different layers. So it really comes down to the question of interest, and each imaging modality or multiple imaging modalities may be able to answer the same question or complement each other.
Thank you, Dr. Lee. We have great questions coming in, but I believe this is all we have time for. We have one question left. Using the Lumina 3, can you image both the tumor as well as the trafficking of the immune cells, CD8, simultaneously using two bioluminescent signals?
The quick answer to that is absolutely yes. If you want to look at two different populations at the same time, there's multiple ways of doing that. Simply, you do need to express two different reporter genes. So, for example, you could do a firefly luciferase and a ranula luciferase. The way that works is that each luciferase interacts with a different substrate. So if that substrate is not present, then that luciferase should not emit any signal, even though that protein is being expressed. So with firefly, let's say firefly for the tumor and ranula for the immune cells, you can inject luciferin, or actually, take that back, you can inject the ranula substrate called solentrazine, which undergoes flash kinetics, and that allows you to actually see the immune cells relatively quickly. And because it undergoes flash kinetics, that signal will dissipate in the order of maybe even an hour or 2 hours. And then you can inject the luciferin and be able to see the tumor population itself. Alternatively, what you can do is do what's called spectral mixing, which is that each of these different luciferases emit light at different wavelengths. So you could acquire the data at the same time and then spectrally, post-acquisition, unmix those two signals based on wavelengths.
Thank you, Dr. Lee. I would also like to thank Dr. Bernthal for his presentation as well. Dr. Lee, would you like to leave us with any final comments for the audience?
No, I'd like to thank everyone for attending, and hopefully it was an informative session for everybody. And also, if you can push out the link for the second link that we have there to the audience. That's our website for our institute, and I think it offers enough information about what we discussed today. But also, there's a link there for some applications that you can review what types of publications and research people have done with different types of imaging modalities that are available that we discussed today.
Thank you again, Dr. Jason Lee and Dr. Nick Bernthal. I would also like to thank our sponsor, PerkinElmer, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through June 1st, 2017. You will receive an email from Labroots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you so much for joining us, and we hope to see you next time. Goodbye.